Welcome back, everyone, from the break. Hopefully, everyone was able to take a moment to refresh. We will now continue with our next session featuring speakers from the Center for Biologics Evaluation and Research, or CBER. Please join me in welcoming our presenters. First, we'll hear from Lonnie Warren Henderson, Public Affairs Specialist within the Manufacturers Assistance and Technical Training Branch in the Division of Manufacturers Assistance and Training within CBER's Office of Communication, Outreach, and Development. The presentation will be followed by Heather Erdman, Associate Director of Quality Assurance in the Office of Review Management and Regulatory Review in the Office of Therapeutic Products within CBER. Then we'll have a presentation by Dr. Karen Knudsen, a CMC reviewer in the Office of Cellular Therapy and Human Tissue CMC, also within the Office of Therapeutics within CBER, followed by a presentation by Dr. Devavina Day, a Pharmacology Toxicology reviewer in the Office of Pharmacology Toxicology in the Office of Therapeutic Products, also within CBER, and then followed by a presentation by Dr. Jessica Lee, Branch Chief for the Oncology Branch 2 in the Division of Clinical Evaluation and Oncology, also within CBER. And now I'll turn it over to our first presenter, Lonnie Warren Henderson.
Thank you so much. I am happy to be here this afternoon to provide a quick overview of CBER's Manufacturers Assistance and Technical Training Branch that I will refer to as MAT-B during this presentation. During this presentation, I will discuss who we are, to include our mission, talk a little bit about what we do, highlight some do's and don'ts concerning the types of inquiries with which we assist, and sum things up with a take-home message and contact information. Who are we, you might ask? Well, let's take a look at where we are within CBER's organizational structure. MAT-B is located within CBER's Office of Communication, Outreach, and Development under the Division of Manufacturers Assistance and Training. Our mission is to provide our stakeholders with access to timely, accurate, and useful information about CBER-regulated products and services. What we do. We respond to industry inquiries. We coordinate liaison meetings with industry. We coordinate public events such as workshops and conferences. We have an exhibit program, and we coordinate and track requests for CBER speakers. For the purposes of this presentation, I will focus on inquiries and liaison meetings. MAT-B receives questions from all facets of industry via telephone and email, to include big pharma, small startups, blood banks, and tissue banks, to name a few. Oftentimes, we are the first point of contact with FDA. It is also important to note that we still answer phones live and that we do not have a phone tree system. We are very proud of that fact. We respond to general information requests from industry received by email or phone concerning CBER's policies, procedures, and regulations. We provide this information using publicly available resources found on FDA's website and more specifically from CBER's web pages. We often respond in real time by navigating inquiries to the information over the phone. However, due to the complex nature of biological products, we also at times have to consult with subject matter experts from our product offices. If your questions do not pertain to a CBER product, we will redirect your inquiry to the appropriate center or FDA component. All centers work together to ensure that the inquiry is properly handled. After all, we are one FDA. Here are some do's and don'ts with regard to inquiries. You may contact MAT-B if you need assistance prior to submitting an application. You may contact MAT-B if you are a lawyer or consultant representing a firm. However, please do not contact MAT-B if you have already had a meeting with FDA about your submission. We recommend that you contact the product office. Do not contact MAP-B if you are a lawyer or consultant that is not representing a firm. We recommend that you contact our Consumer Affairs branch. Please note that MAP-B will assist if you need help with finding contact information for your assigned regulatory project manager or any other contacts within the center. Another important function of MAP-B is to help coordinate liaison meetings between our product offices and industry organizations. You can find more information about liaison meetings in our SOPP 8101.2. These meetings are periodic meetings between CBER and an external organization representing a group of interested parties. The meetings are meant to provide an opportunity to discuss topics of mutual interest to CBER and the organization. For example, we've had liaison meetings with the American Association of Tissue Banks and the Biotechnology Innovation Organization. These meetings occur at the request of the external organization, and MAP-B serves as the point of contact. Liaison meeting summaries are available via a Freedom of Information Act request. In summary, we are here to help. After all, assistance is our middle name. We focus on outreach through many of our functions, and we are only a phone call or email away. And finally, here are some important contacts. Thank you so very much for your attention, and I am happy to answer any questions later.
Thank you, Lonnie, for that introduction. I'm Heather Erdman with CBER's Office of Therapeutic Products, or OTP. I'll be providing a high-level overview of the regulatory resources and avenues for obtaining early guidance from CBER OTP, including meetings, web pages, webinars, and additional resources. The objectives of this presentation include facilitating your ability to locate resources for interacting with CBER OTP, to understand communication, submission, and meeting options for early interaction with OTP, to enhance your understanding of the processes through OTP Learn, and to recognize additional regulatory resources for your future efforts. So, who is CBER OTP? CBER OTP is one of 3 CBER product offices responsible for the regulatory oversight of biological products, including purified and recombinant proteins for hematology, antivenoms, gene and cell therapies, therapeutic tissue engineered products, human tissue products, therapeutic vaccines and antigen-specific active immunotherapies, certain medical devices, and xenotransplantation products. Sponsors of products regulated in OTP have several opportunities to interact with the office, which may include formal and informal meetings. Submissions may be sent to CBER's Document Control Center per the guidelines detailed on the web page entitled Regulatory Submissions in Electronic and Paper Format for CBER Regulated Products. These guidelines will address the various forms and methods by which a request may be submitted. When emailing, secure email policies should be followed per CBER SOPP 8119. Upon receipt of a submission, an assigned Regulatory Project Manager, or RPM, will serve as a liaison between the requester and the FDA review team. Activities related to requesting, scheduling, conducting, and documenting formal meetings are defined in FDA regulations, guidance documents, and SOPs. CBER OTP offers a comprehensive set of web pages to guide sponsors on these processes. It's entitled Interactions with Office of Therapeutic Products. This website and this presentation will focus on biological products regulated under an IND and BLA pathway. For similar information for OTP-regulated medical device products, please refer to the Guidance for Industry entitled Request for Feedback and Meetings for Medical Device Submissions, the Q Submission Program. This comprehensive website, the Interactions with OTP, provides resources for the potential meeting types applicable throughout the product lifecycle, including early interactions such as interact meetings and pre-IND meetings, in addition to meetings at the investigational, marketing application, and post-marketing stages. These include meeting types A, B, C, and D, as well as requests for clarifications for any of these meetings. Formal meetings may be in a face-to-face, teleconference, or written response only, or WRO format. Face-to-face meetings include in-person meetings and virtual teleconferences on IT platforms that allow for both audio and visual communication. During this presentation, I will focus on early interaction meetings such as Interact and pre-IND meetings, and briefly touch on the other meeting types. Initial targeted engagement for regulatory advice on CBER products, or Interact. These Interact meetings provide sponsors that are developing novel therapies an opportunity to request feedback at an early stage of development. OTP will determine whether the status of the product development is premature or too advanced for an Interact meeting. The sponsor should have identified the investigational product planned for a clinical study and conducted preliminary preclinical proof of concept studies. The sponsor should not have conducted definitive toxicology studies at this stage. This website will detail OTP procedures following the receipt of an Interact meeting request, the criteria to consider in a complete and informative meeting request and meeting package, the timelines, and common reasons for Interact meeting request denial, such as stage of development or meeting package deficiencies. A Type B pre-IND meeting provides a sponsor the opportunity to obtain feedback on their product development program that may not be answered by guidance documents and other publicly available resources. Information obtained from a pre-IND meeting can assist sponsors in preparing a complete investigational new drug or IND application and potentially reduce the risk of a clinical hold. The sponsor may seek feedback on the design of preclinical studies and the initial IND study and product manufacturing and quality controls needed to initiate human studies. The meeting also provides an opportunity to discuss plans for studying the product in a pediatric population and the best approach for presenting the data in an IND. There are different categories of meetings, including Type A, which are reserved for stalled product development programs, Type B, Type C, and Type D meetings. Type B meetings may include, but not be limited to, an IND end-of-phase meetings to review the development program, a Type B pre-BLA meeting to discuss the proposed content of and readiness for Biologics License Application, or BLA, and identify any major unresolved issues from the development program, and also for products developed under Breakthrough Therapy designation and or Regenerative Medicine Advanced Therapy, or RMAT designation, sponsors receive more intensive guidance on a development program with increased interactions with OTP. Type C meetings are another meeting category where the request does not quite fit in a Type A or Type B category. And lastly, Type D meetings may be requested at any stage of development and should be limited to 1 or 2 focused topics, not exceed 3 to 5 questions, and should not require input from more than 3 disciplines. A request for clarification may be sought after a Type A, B, C, D, and Interact meeting to ensure the requester's understanding of FDA feedback provided in either a preliminary response if the meeting was canceled, a meeting summary, or a written response issued by FDA. Only questions of a clarifying nature should be submitted rather than new issues or new proposals. Extensive guidance is provided on the web pages, including procedures, unique timelines, meeting package expectations, and best practices for managing each type of meeting with OTP. There are additional opportunities to engage with OTP, which I will not address in detail here, but will list out for your reference. One includes the CBER Advanced Technologies Team, or CAT meeting, which may be granted to prospective innovators and developers of advanced manufacturing and testing technologies to discuss the novel technology rather than a specific therapeutic product. OTP also provides multiple resources for your ready access, including OTP Learn, which currently provides 30 webinars. These trainings include, but are not limited to, original IND content and format, which includes best practices for generating cover letters, a CMC section of a gene therapy IND, and early phase trials for cellular and gene therapy. The webpage entitled OTP Events, Meetings, and Workshops provides links to 22 recordings. Included are the OTP Town Hall series, designed to engage with product developers and researchers to discuss topics related to OTP-regulated products. The Town Halls have a question and answer format designed to provide regulatory information to advance drug development. A recent topic includes non-clinical assessment of cell and gene therapy products. Also, RegenMed Ed, an FDA CBER OTP series on regenerative medicine, brings together stakeholders and OTP staff to discuss information about regenerative medicine therapies and explore opportunities for patients, caregivers, and advocates to engage with FDA to help advance product development. A recent topic includes clinical trials, the patient experience. In addition to our event series, OTP also hosts several meetings and workshops throughout the year. Most of these events are held virtually and are free and open to the public. This cellular and gene therapy guidances link outlines 36 guidances for industry and in some cases links to a related webinar. For general regulatory support and procedural questions, please feel free to email otprpms@fda.hhs.gov. A regulatory professional will respond and, when needed, engage some review or policy personnel to gather a response for your question. It is important to note, though, that this email address is provided to support questions pertaining to products related under CBER OTP only. Provided in the next 3 slides are resources for your ready reference, including references cited in the presentation and supporting the topics covered. Offered here are meeting-related resources. Note that within the Interactions with OTP webpage, there are links to additional web pages detailing each type of meeting and the respective requirement and recommended best practices. Listed here are guidance documents supporting various phases of product development and FDA engagement. Additional resources are provided here for trainings, webinars, workshops, and additional guidance on how to submit submissions to CBER. There are many opportunities to interact with OTP throughout the product lifecycle, and early and timely engagement is recommended. OTP provides numerous online resources to help enhance your understanding and facilitate your interactions with OTP. We look forward to working with you. And now I'd like to turn this over to Dr. Karin Knudsen. She'll be covering the CBER's CMC considerations for early phase studies of cell and gene therapy products.
Thank you, Heather. So as Heather stated, my name is Karin Knudsen, and I am a Chemistry, Manufacturing, and Controls Reviewer, or CMC Reviewer, within the Office of Therapeutic Products in CBER. Today, I will be talking about CMC considerations for early phase studies, specifically for cell and gene therapy products. So by the end of this presentation, you should understand why manufacturing investigational cell and gene therapy products presents unique challenges. You should be able to identify the CMC information you need to include in your early phase IND submission, and you should be able to describe some common CMC issues that arise during product development and also identify some approaches for avoiding them. So within OTP, we regulate a diverse product portfolio, which includes gene therapy products such as genetically modified cells like CAR T cells, viral vectors, and tumor vaccines. We also regulate cell therapy products like stem cells, anti-tumor and antiviral T cells, and xenotransplantation products. The diversity of products we regulate means that we encounter different manufacturing paradigms than typically seen with conventional drugs or biologics. With conventional drugs or biologics like vaccines or monoclonal antibodies, typically one product lot is manufactured to treat many patients. But in the case of cell and gene therapy products, we usually encounter the situation that one product lot treats a few patients, or in some cases, one product lot will be manufactured to treat only a single patient. This difference means that sponsors will encounter many unique issues during the development of these products, including the use of advanced manufacturing techniques, issues with scale-up of manufacturing and distribution, and typically the impact of a manufacturing failure is greater for cell and gene therapy products. Because of the diversity of products we regulate and the different manufacturing paradigms and issues we encounter, the regulatory review of cell and gene therapy products is highly product dependent. For example, for a specific type of product, one lot could treat thousands or just one patient. Sponsors use a wide variety of manufacturing procedures and techniques to manufacture their products. We also see that products that have little test material or a short shelf life will present challenges with comprehensive testing, and that is generally handled on a case-by-case basis. I do want to make the point that the risk of each product varies because that risk is dependent on the source material and how the product is made. Finally, some of our products are highly variable, especially in the case of an autologous product, which makes demonstrating manufacturing consistency and comparability challenging. That again will need to be handled on a case-by-case basis. So given that the regulatory review of cell and gene therapy products is highly product dependent, how do you actually develop your products, and what is paramount to tackle early to avoid review issues? So overall, the stage of product development guides the review concerns, but safety is the primary concern at all stages of the product development lifecycle, regardless of whether you're coming in for a phase 1 study or submitting a biologics license application. As you progress through the product development lifecycle, you will need to start focusing on different aspects of product development, such as product characterization, qualification and validation studies, and assessing product potency. But overall, product development does tend to occur on a continuum, and as you learn more about your product during clinical studies, our expectations will increase. I do want to emphasize that product safety and quality do not happen by chance. Safety and quality need to be designed into the product, so you should start this process early. It is especially important that you start product development early because cell and gene therapy product development should progress in parallel with clinical development. We have seen sponsors struggle to keep up the pace of their CMC development compared to their clinical development, which we all know can happen very rapidly nowadays. Just to reiterate, CMC expectations do increase through the product lifecycle, so you need to stay on top of this. But today, we're focusing on early stage INDs, so let's discuss what chemistry, manufacturing, and controls actually are. So chemistry, manufacturing, controls, or CMC information covers everything about how you manufacture your product, and it should describe how you ensure the safety of the source materials, intermediates, and final product. So this information includes details on product manufacturing, details about the raw materials you use, the manufacturing process and the quality systems, and product safety and quality testing, either in process or release testing. This information also includes product stability, storage and shelf life, what container you use, what label you use, how you track your product. In some cases, you may be able to cross-reference related INDs or master files to provide some of this information, but documentation of that cross-reference is needed. Overall, the CMC information provided in your IND should demonstrate your capability to consistently and reproducibly manufacture the investigational product. It should include information that describes the composition, manufacture, and control of the investigational product, and it should be sufficient to assure identity, quality, purity, and potency or biological activity of the investigational product. So diving more into this, as stated earlier, early phases focus more on safety and specifically knowing the identity, purity, and activity of your product. The amount of information that you will need to submit under your IND will depend on the phase and the scope of the initial clinical investigation. And as development proceeds, it will be necessary to supplement the initial CMC information to address the expanded phase and scope of your clinical investigations. And as you move on to later phases, we will require more information on the quality and potency of your product. So what are some of the common issues that we see with early phase studies, and how can you avoid these CMC issues with your submission? Remember, we are primarily focused on product safety here. If you are using donor-derived materials, you should describe how donors are tested to ensure the source material is safe. Sometimes sponsors don't include sufficient information about the release assays that they perform to ensure safety of their product, so make sure you include comprehensive information about your release assays. In particular for safety assays, such as those assays for sterility and mycoplasma, You should describe the methodology, and in general, safety assays should be qualified for their intended use for early phase studies. Determining the dose of your product is really important, so you may need to qualify that assay for its intended use. We also see that some sponsors don't include sufficient information about the reagents they use during manufacturing. You should include the source and documentation for each reagent, which is typically provided as a certificate of analysis, and that information will be used to show that each reagent is suitable and safe for use. One final consideration I will mention is that you should document the delivery device you intend to use to administer your product, including its regulatory status, and provide data demonstrating that your delivery device is compatible with your drug product. While I talked about quite a few safety issues you should consider and address in your IND, please be aware that this is not an all-inclusive list. I highly recommend that you read the FDA guidances about the CMC information you need to include in your IND. There is a gene therapies guidance and a cell therapy guidance, and both contain very helpful information. But what should you do if you have more questions? If you have more questions for us, I highly recommend you take advantage of the meeting opportunities that Heather went over in her presentation. In particular for early phase studies, the Interact meeting and the pre-IND meeting should be used to discuss any unique issues you are running into or any questions you may have regarding your product prior to submitting your IND. There are other meeting opportunities for later phase studies, but since this is an early phase study discussion, please consider an Interact or pre-IND meeting. So finally, to summarize what we talked about today, the CMC information submitted under early phase INDs should demonstrate your capability to consistently and reproducibly manufacture a safe investigational product. The regulatory review of cell and gene therapies is highly product dependent, so we do recommend that you talk to the regulators, which is FDA, about challenging issues or novel approaches you plan to use. Safety and quality must be designed in the product, so I'm going to stress again, start this process early. This slide provides contact information and a lot of links to websites that contain valuable information on CBER INDs and how you can interact with us. That concludes my presentation. I will now pass it on to Divavina.
Thanks, Karen, for your overview of CMC considerations for cell and gene therapy products. This talk will focus on the non-clinical assessments of CGT products to support an IND. What I hope you will learn from this talk are the regulations that mandate non-clinical testing, what are the non-clinical considerations for early phase trials, links to relevant guidance documents, and a discussion on when to have interactions with the FDA for your non-clinical program. Title 21, Part 312, Section 23A8 of the Code of Federal Regulations mandates non-clinical testing. This regulation outlines the details of pharmacology and toxicology assessments of a new drug required as part of an investigational new drug or IND application. It is important to note that the kind, duration, and scope of such assessments varies with the nature of each proposed clinical investigation, implying that the non-clinical evaluation for each product is determined on a case-by-case basis. In the life cycle of a drug development, non-clinical studies typically come in at early stages before initiating the phase 1 clinical trial. What sort of data can support the initiation of a phase 1 clinical trial? This can be data derived from multiple sources, such as GLP or non-GLP, well-controlled non-clinical studies, published data in peer-reviewed journals, cross-reference to similar products in previous submissions, as well as data from clinical trials, can all be used to support an IND. The primary expectation from a non-clinical program is to demonstrate the safety of the CGT product through well-designed toxicology studies.
The goal of toxicology studies is to determine the no observed adverse effect level, or the NOEL as it is called, for the product, and evaluate any unintended activity of any component of the product, such as the cells, vector, transgene, or packaging elements. Such unintended effects could include presence and activity in any off-target tissues, any local or systemic effects seen either acutely or in the long term, potential risk of immunogenicity or tumorigenicity from the product, or any unintended effects of the drug delivery systems used. While we are on toxicology studies, I do want to emphasize that as per 21 CFR Part 312, each toxicology study should be performed per Good Lab Practices or GLP. If this cannot be done, an explanation should be provided, and all non-GLP studies must have an oversight by an independent quality assurance person or team. In addition to demonstration of product safety, the non-clinical program is also expected to demonstrate the activity of the CGT product in vitro and, when feasible, in vivo in a relevant animal species or any other alternate test systems. Data from such proof of concept studies, along with findings from toxicology studies, should together inform about the optimal dose levels and dosing regimen to be used in the proposed clinical trial. Cell and gene therapies have several potential safety concerns, which can either be associated with the product itself, as listed on this slide, or the procedure that is used to deliver the product. Ideally, the product administered in non-clinical studies should be identical to or as similar as possible to the intended clinical product. For any animal-derived analogous products, it is important to characterize the level of analogy between the human and the animal product and clearly justify the human equivalent dose or HED calculations. It is critical to use an appropriate animal model for non-clinical studies. The animal model used should be permissive to the product and/or recapitulate elements of the target indication. Therefore, there is no default animal model, and scientific justification should be provided for each species or model used. This slide gives some pointers to help select the most appropriate animal model to assess the safety and activity of a specific CGT product. We will now spend a few minutes on non-clinical study design considerations, which is the key to get accurate results from non-clinical studies. A robust study design is non-biased, mimics the planned clinical scenario as closely as possible, includes appropriate controls and multiple dose levels of the investigational product, and includes adequate animal numbers in each study group, all while incorporating the 3 R's for each study. A robust study design is also of a sufficient duration, incorporates multiple time points for in-life assessments, and includes multiple assessments, both in-life and at necropsy. Such a robust study design is key in accurate evaluation of the long-term safety and activity of a CGT product. Some of the key assessments of safety and activity of a CGT product which should be tested at multiple time points include functional outcomes, kinetics of biodistribution, tumorigenicity, transgene activity, and immunogenicity. Toxicology assessments include mortality, any clinical changes including body weight, and a comprehensive evaluation of adverse findings in each animal in each study group. As discussed by Heather and Karen, we encourage sponsors to communicate with the FDA from the earliest stages of product development via the Interact and pre-IND meeting pathways. From a non-clinical standpoint, an interact meeting would be at a stage when the sponsor has generated preliminary safety and proof of concept data, but has yet to initiate definitive safety studies. Guidance from CBER at this stage should help the sponsor design robust safety and toxicology studies for their specific product. The goal of a pre-IND meeting, on the other hand, is to achieve a successful IND submission. From a non-clinical standpoint, this is a stage when all proof of concept and preliminary safety studies have been completed, and the sponsor is ready to conduct definitive safety studies. A pre-IND package should discuss all the elements of a non-clinical study design that was discussed previously, such as animal models used, dose, regimen, endpoints, sacrifice intervals, the rationale for using each of those elements, and the resulting data interpretation. To summarize, the non-clinical program for any CGT product is determined on a case-by-case basis. Non-clinical data submitted in the IND should support the safety and biological activity of the CGT product in the proposed clinical indication. There are multiple opportunities to obtain early feedback from the FDA on non-clinical development plans prior to IND submission. It is important to note that novel therapies mean novel testing paradigms, and hence pre-submission interactions with the FDA is highly encouraged. So come to us early. These are some relevant guidances to help design non-clinical studies for CGT products. And here is my contact information if you have any questions about this talk. Thank you all for your attention.
Hi, this is Jessica Lee. I'm the Branch Chief of Oncology Branch 2. Today, I will present early development of cell and gene therapy in oncology, clinical consideration. My disclosure. My presentation will cover key clinical regulatory issues frequently encountered during early clinical development of cell and gene therapy in oncology. Outline of today's presentation. First, I will provide a high-level overview of early clinical development of cell and gene therapy in oncology, followed by discussing key clinical considerations, including trial design, endpoints, dosing and dose escalation, and safety. Last, I will conclude with a brief summary. Overview. In early phase cell and gene therapy trials, several key factors are crucial, including number 1, safety. The primary objective of early phase cell and gene therapy study usually focus on demonstrating the safety of the investigational product. Number 2, selection of the starting dose for first-in-human study. Due to the lack of clinical data, selection of starting dose for first-in-human trial will be based on extrapolation from animal studies. Number 3, dose exploration, which usually varies according to different products. In addition to determining the maximum tolerated dose, additional dose exploration strategy includes to determine the optimal dose for safety and efficacy, and to determine the feasible dose that can be consistently manufactured and administered. Number 4, activity assessment. In addition to safety and dose determination, consider to evaluate preliminary clinical efficacy and to determine if there is signal for potential benefit. Number 5, manufacturing feasibility. Due to the complex and challenging manufacture process for cell and gene therapy product, consider incorporating assessment of manufacturing feasibility into study objective. In the next section, I will provide high-level coverage on study design, endpoints, dosing and dose escalation, and safety. First, study design. Most of the first-in-human cell and gene therapy trials in oncology are single-arm studies and often focus on unmet medical needs, such as patients with disease relapsed or refractory to available therapies. However, for combination therapy, it will be challenging
to assess the contribution of effect in a single arm setting. The mechanism of action for cell and gene therapy often involves specific targets. Therefore, may require companion diagnostics to identify suitable patients. Depending on targets, consider reaching out to either CDIH or CBER OBRR to validate the in vitro diagnostic assays. In addition, companion diagnostic assays may require a protocol-specific risk evaluation. For significant risk devices, IDE are required before the study can proceed. Study endpoint. When defining study endpoints in cell and gene therapy trials, consider the following. As mentioned before, most of the first-in-human cell and gene therapy trials in oncology are single-arm study, with main purpose to demonstrate the safety and to find the optimal dose for the investigational agent. Other key endpoints include tumor response rate and duration of response. There is limitation of single-arm trial in that tied to event endpoints, including overall survival and progression-free survival, can be challenging to interpret without concurrent control. When assess the endpoint, consider the impact of concurrent treatments. Patients might receive combination regimens such as lymphodepletion chemotherapy, interleukin-2, or combination with other immunotherapy, including immune checkpoint inhibitors. These factors can confound the results and complicate the evaluation of the investigational product's true effect on safety and efficacy. Dosing and dose escalation. When selecting the starting dose for early phase and first-in-human study, please provide a robust rationale and justification for the selected starting dose based on preclinical data and any clinical data from similar products of the same class. In general, selection of starting dose for first-in-human study is usually based on toxicology data and prior human experience if similar constructs have been used in previous studies. For engineered cell therapy, the dose should be based on the number of transduced cells rather than total cells. In general, dose escalation schema for early phase cell and gene therapy trials should consider anticipated cell expansion in vivo and anticipated toxicities. In addition, for cell and gene therapy, we generally recommend a half-log increment between dose levels. Regarding dose escalation schema, both the 3+3 design and Bayesian's adaptive designs are acceptable. In general, we do not recommend intrapatient dose escalation for cell and gene therapy. Last, consider implementing staggered dosing between subjects within each cohort and between cohorts. Safety. The main purpose of early phase and first-in-human trial is to demonstrate the safety of the investigational product. Ensuring safety in clinical trials involves several critical components, including but not limited to safety monitor and management plan, dose-limiting toxicity criteria, and stopping rules. Of note, the safety monitor and management plan should include duration of safety monitor, management plan for potential toxicity, and monitor for potential adverse events of special interest, if any. Now I will elaborate further on safety monitor plan, focus on duration of monitor and long-term follow-up plan. Regarding duration of monitor for adverse events, safety monitor should extend long enough to cover the expected duration of the therapy's effect. In addition, duration of monitor will depend on preclinical studies and prior safety information of related products, if any. Regarding long-term follow-up, the follow-up duration should be tailored to individual product. For example, certain cell and gene therapies, particularly those involving integrating viral factors, may require extensive follow-up up to 15 years to fully assess long-term safety and late onset effect. Dose-limiting toxicity. Regarding DLT observation period, ensure sufficient observation period to cover both acute and subacute toxicities. Please note concurrent treatment can confound and complicate DLT assessment. In general, DLTs are context-dependent and may vary by therapy, such as cytokine release syndrome and off-target toxicities. Please provide clear definition for DLT. Use established criteria like CT-CAE or AST-CT to define DLTs to ensure consistency and accuracy in evaluation. For example, for cell and gene therapy study in oncology, DLT often include grade 3 or higher major organ toxicity, provided these are not pre-existing or not related to the underlying malignancy. Trial stopping rules. In general, trial stopping rule will trigger temporary pause in enrollment and treatment to limit exposure to excess risk. This pause is not meant to terminate the study, but to allow for thorough safety assessment. Specify well-defined conditions such as type and number of adverse events that trigger a temporary suspension of enrollment and dosing until safety review is completed. Based on the safety assessment, protocol revision may be needed. This could include changes to eligibility criteria, dosing regimen, or the safety monitor plan to mitigate identified risk. Consider include deaths not due to progressive disease within 30 days of receiving investigational cell and gene therapy, or any deaths directly attributed to cell and gene therapy as trial stopping criteria. In summary, cell and gene therapy hold significant promise in cancer treatment, with products advancing rapidly from bench to bedside. These therapies are complex and diverse, with unique mechanism of action and inherent safety concerns. Therefore, each clinical trial must be evaluated individually, considering the specific characteristics and the risk of the therapy. Thanks for your attention. This concludes my presentation. Here is a list of useful FDA resources. This is a list of FDA contact information. The next section, I will be happy to answer any questions you might have.
All right, thank you again to our presenters for their fantastic presentations. We will now begin our question and answer panel. Joining our speakers for this panel is Dr. Peter Bross, Chief of the Oncology Branch of CBER's Office of Therapeutic Products. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Now we'll start with a question that came in for Heather Erdman. Is it difficult to get an Interact meeting granted by the FDA?
That is a good question. It is not difficult to get an Interact meeting granted as long as the criteria of an Interact meeting is followed. The stage of development and the meeting package, format, and content, including how the direct questions are posed, have an impact on this decision. Using the direction provided in the Interactions with OTP webpage and the related guidance document in SOPP 8101.1 can provide that specific guidance that you can consider for your current need. Thank you.
Thank you so much for that response. Another question for you. Can a Type D meeting be requested as the first meeting with the agency? Sometimes there are simple questions that would be nice to be answered without the detailed background needed for an Interact or a PIND.
That's another great question. Yes, a Type D meeting may be considered for a first meeting, again, as long as the criteria of a Type D meeting is met, such as no more than 1 to 2 topics with no more than 3 to 5 questions posed, including a restricted number of disciplines. Please note that a limited number of Type D meetings may be granted in a close period of time, and these could be combined to make up one or another meeting type, for instance. Therefore, prospectively planning when and what meetings you want held with FDA aligned with your product development timeline really is ideal. Thank you.
Thank you so much for sharing that information. And now we'll move on to a question for Dr. Karin Knudsen. How much stability data is required for cell and gene therapy IND submissions?
Great, thank you for this question, and it is a great question. So the amount of product stability data required for your product will of course depend on the phase of the clinical study. However, I do want to make the point that stability data is required in all phases under the IND to establish a shelf life for your product and to demonstrate that the product remains within acceptable limits for the duration of your clinical trial. So typically, we expect preliminary stability data, especially for the in-use product. We expect that to be available for the start of your phase 1 study. So you should submit preliminary stability data for your product in your IND, as well as your stability study design plan for long-term stability studies. Thank you.
Thanks so much for that response. We have another question for you, Dr. Knudsen. When does the FDA expect potency assays to be developed and used for release testing during the product's life cycle?
So thank you for this important question. We recognize that potency assay development is generally difficult for cell and gene therapy products given their complexity and at times their inherent variability. So potency does apply at all stages of clinical development, but your potency assay is required to be used during the clinical studies intended to provide the primary evidence of effectiveness to support a marketing application. So this would be your pivotal study, and that is when that potency assay needs to absolutely be implemented. Your potency assays may not be completely defined early in product development because your product and process knowledge is limited, but it should become progressively more comprehensive as you accumulate manufacturing experience. Following up on what I said in my presentation, we highly recommend that you start product characterization and potency assay development early, even during your phase 1 study or during your preclinical studies, so you do not run into issues later during the product life cycle. Not having an adequate potency assay in place is an issue for our sponsors going into their pivotal study. So overall, we recommend that if you do not implement a potency assay for release testing for your early phase studies, you should provide in your IND submission a description of your general plans for product characterization and potency assay development and how you will reassess your potency assurance strategy during product development. Thank you.
Thank you so much for that thorough response. Now we'll have a question for Dr. Devavina Day. Dr. Day, can you compare and contrast the differences in CBER's use of the NOEL or NOAEL versus a MABEL approach?
Hi, thank you for that question. Yes, that's a very interesting and important question. While I should speak to NOEL, which is more for cell and gene therapy, that is the no observed adverse effect level, which is typically the highest safe dose level for a cell and gene therapy product. However, a MABEL, which is traditionally used for monoclonal antibodies, is the minimal dose level at which one sees biological effect. So both look at pretty different aspects. However, I think the important thing is both look at. Thank you.
Thank you so much for that response. We have another question for you, Dr. Day. When you mentioned GLP compliant toxicology studies conducted by a certified testing facility, does FDA certify the testing facility for a GLP laboratory organization? If not, what does it mean by a certified testing facility?
Thank you for that great question. To clarify, FDA does not certify GLP facilities. However, there are multiple and several GLP certification programs and organizations who do the certification, and those are all recognized by the FDA. Some of such certification programs are those issued by the CT, CFPI, and there are several other such programs. However, FDA does not certify any program. Thank you.
Thank you so much for that clarification. And now we have a question for Dr. Peter Bross. We did phase 1 studies in Europe about 10 years ago. We are planning to do phase 2 studies in the USA. Do we have to submit an IND or base our IND in Europe? Can we do phase 2 studies in the USA?
Okay, great question. Can you hear me okay? The answer is that you need to submit an IND to do a study in the US. And we see this more and more frequently that there are clinical development programs which are based on studies initially conducted in different countries, but not under a US IND. So in order to do the studies in the US, you will need a US IND. And if you have already performed clinical studies, I would recommend that you request a pre-IND to discuss, because there may be some differences in requirements for product and preclinical evaluation. And we're seeing more and more of these studies that are clinical development programs that are initiated internationally, and then they come to the US, and we have a specific guidance on that and a specific code of federal regulations. But my suggestion would be to come in for a pre-IND to discuss the requirements.
Thank you so much for that response, Dr. Bross. Another question for you. What is your recommendation on FDA notifications for studies to be conducted internationally? How early should those notifications occur?
Okay, well, this is kind of an interesting question related to the other question, but depends what you mean by notifications. As I mentioned, that in order to do a study in the US, you would need to submit an IND and have the IND be in effect. So the IND needs to be in effect before you do the studies. You can also do foreign clinical studies under a US IND, but it's really just required that you have an existing and open and in effect IND in order to initiate studies. And if you're studying, conducting studies internationally, then it's up to the sponsor when you want to notify us. But I think it's generally a good idea to request a pre-IND after you've had some clinical experience. Thank you.
Thank you again so much for that response. And now we'll have a question for Lonnie Henderson. If we've already met with CBER about our product, can we still call Matt B with a general question unrelated to our submission?
Thank you for that question. Yes, you can call Matt B with a general question so long as it does not have anything to do with your meeting with FDA or any other submission.
All right. Thank you so much for we have another question for you, Lonnie. Sorry, just a moment. If we have an emergency and it is after close of business, whom should we contact?
Thank you also for that question. If you have an emergency, such as an emergency IND request, and it is after close of business, you should contact FDA Emergency Operations, and that information is available on FDA's website. Thank you.
Thanks so much for that response as well. And now we have a question for Dr. Jessica Lee. What is your general approach for grade 5 or death toxicities after receiving investigational cell and gene therapy products?
Thanks for the question. This is a great and important question. As pointed out in my presentation, the main purpose of early phase, including first-in-human cell and gene therapy study, is to demonstrate the safety of the investigational product. Therefore, we usually take every grade 5 toxicity, which is death, very seriously. In general, we will carefully review the product certificate of analysis and the clinical data, which will include eligibility criteria, the actual dose received, and the comprehensive medical record focused on baseline and post-treatment clinical information, and the autopsy result if available. Thereafter, based on the outcome of the safety assessment and the identified risks associated with the treatment, we will usually consider amending the protocol to revise trial design, eligibility criteria, dosing regimen, DLT criteria, stopping rules, safety monitor management, and supporting care to mitigate the risk associated with the investigational treatment. Thank you.
Thank you so much for that response, Dr. Lee. We have another question for you as well. Could you please discuss further on the long-term follow-up plan for CGT study?
Yeah, thanks for the question. Each cell and gene therapy are unique, and many require long-term follow-up. In general, the requirement and duration of long-term follow-up will depend on the product. And we usually adopt a risk-based approach to determine the requirement and duration of long-term follow-up. There are several factors taken into consideration, which include whether the manufacturing of the cell and gene therapy product involves gene editing or integrating a viral vector, whether there's long-term expression of the vector or latency or persistency or reactivation. For example, we generally recommend up to 15 years for gene editing product and up to 15 years of integrating vector, such as retrovirals, lentiviral based, and transposon element. And up to 5 years for AAV, which is non-integrating but with long-term expression. So in general, this will be based on the product and will be considered a case by case. Thank you.
Thank you again for your response, Dr. Lee. Now we'll address a question that came in for Dr. Karin Knudsen again. Does the potency release assay need to be qualified? If yes, when?
Yes, I am happy to address this follow-up question. So yes, very short answer, yes, your potency assay will need to be qualified. The longer answer is that it will need to be qualified by the time you start pivotal studies. However, even if qualification of the assay isn't required, we generally recommend that you evaluate assay performance at all stages of development, because assays that don't measure what they're intended to measure, or assays with high variability or little robustness, will provide you with little to no information regarding meaningful control of your product. If you do have additional questions about potency assay development, qualification, or validation, I recommend you search for our potency guidances. There's actually a new draft guidance from last year, from 2023. I also recommend that you come in for a meeting with us if you run into issues with the development or the qualification or validation of your potency assay. Thank you.
Thank you so much for that response as well. We may have time for one more question. This question came in for Dr. Devavina Day. So GLP animal studies were conducted by another sponsor, and will be cross-referenced by the new sponsor. Does that mean that a new sponsor is not eligible for the Interact because some GLP studies have been completed?
Hi, thank you for that question. In short, no, one can definitely apply for an Interact meeting, even though they do have some data which can be cross-referenced. So yes, for an Interact meeting, typically sponsors do not have data. However, if somebody can cross-reference data which applies to that particular program, you can certainly come in and apply for an Interact and refer to this existing data. So yes, you will absolutely be encouraged to do that. Thank you.
All right, thank you so much for that response, Dr. Day. And looks like that is all the questions we have time for in this session. Thank you again to all our presenters and panelists for all the excellent information. Now we'll take another short 10-minute break and we'll resume at 1:50 Eastern Time.